Hello champions, am I audible? All good, all good. Come fast. Children, children, children. Just give me a thumbs up if I'm audible. Yes. So let's start. Okay. Ready? 1, 2, 3, go. So here I am. I will be only giving the answers, so just understand the concept, okay, along with me. So select one word for the statement. When F1 resemble both the parents, when both the parents are there, what do you call this? You call this as codominance, okay? Wait. You call this as codominance. If F1 resemble both the parents. Now, if F1 does not resemble either of the parents, what do you call this? You call this incomplete dominance. And if F1 resembled either of the two parents, only one of the two parents, that means one parent is dominant, so that is known as dominance. That would be known as dominance. Got it? So this is codominance. This is what? Incomplete dominance, and this would be dominance. Going ahead with the next question. Which of the following is related to hemophilia? My dear children, whatever you remember about hemophilia, you have to talk about that. So it is a autosomal recessive disorder. Okay, always remember it is what? A recessive gene disorder which is there on the, not autosomal, sorry, it is sex linked. It is what? Sex linked recessive disorder. Now, who are more affected? It would be always males who are going to be more affected than females. And what exactly happens over here? A clotting factor is not being produced in this particular case, and hence the person would keep on bleeding. Okay, works. So it is males who are affected both in hemophilia and also color blindness, and both of them are sex-linked recessive disorder. Please remember that. Okay, there is no sex-linked dominant disorders in your NCERT, so please remember that thing. Now, what happens in sickle cell anemia, my dear children? There will be elongated, sickle-like cells, right? And what else happens over here in sickle cell anemia? Basically, initially you had GAG, but it got replaced with GUG. And where is it happening? On the 6th codon it is happening. So initially it was coding for glutamic acid. Now it is going to code for what? Valine. It is going to code for valine. And hence, what is going to change? The beta globin. The beta globin structure is going to change. Hence, the hemoglobin is also changed. Hence, the RBC carrying it, its structure becomes what? Sickle cell. What kind of disease is it? It is what? Autosomal recessive disorder. Again, I am telling you, in your book, you have almost 3 diseases: sickle cell anemia, thalassemia, and phenylketonuria. All of them are autosome linked recessive disorder, not dominant. Only one dominant disease is given, and that too under the pedigree chart, and that is myotonic dystrophy. Please remember that. Rest all of them are recessive. Okay, works? So children, I know that you are texting me. I cannot read it because we are going on really fast, but I just want to help you well, yeah? So which of the following condition correctly describes the manner of determining the sex in the given example? XO type of sex chromosome determine male sex in grasshopper. Children, listen to me carefully. I am going to talk about all kind of sex determination, okay? So in the case of insects, you have X and O condition. Whether it's X chromosome coming from one parent, from the other parent, you don't get anything, which is why this would be what? A male. Always remember, in the case of insects, XO condition is there, yeah, and XO means that would be a male, my dear children. Going ahead, if we talk about birds, okay, in the case of birds, you have ZZ, ZW type of sex determination. What do you mean by ZZ? ZZ means it is male. What do you mean by ZW? It is going to be a female. In the case of birds, they show female heterogamity. That means females are going to produce 2 type of gametes. One is this, the other one is this. Okay, rest all the cases, males are the one that produces 2 type of gametes. Even in the case of human beings, X and Y. Even in the case of insects, X and O. Okay, now there is one more type of sex determination, that is haplodiploid sex determination. That means that is seen in what? Honeybees, my dear children. In the case of honeybees, you have haplodiploid. Always remember H for honeybee and H for haplodiploid. Okay, where the haploid ones are going to be males. Haploid ones are going to be males, and the diploid ones are going to be females. Worse. And now, last but not the least, you have Drosophila. Drosophila is just like human beings only. So it will be again XX, XY type of sex determination. So Drosophila and human beings, birds are the only example of female heterogamity. And in the case of insects, you have XO type of determination. And in the case of honeybees, you have haplodiploid type of sex determination. Okay, got it? Chalo. In a dihybrid Punnett square, how many non-parental phenotypic combinations are present? In a dihybrid, my dear children, you have at the end, after what do you call F2 generation, you have 9:3:3:1 ratio, right? So 9 of them are going to show the dominant parent, that is round and yellow, okay? And 1 of them are going to be showing the recessive parental type, that is wrinkled and green. So this is going to be round and yellow, okay? And this is going to be wrinkled and green. What about them? They are going to be the new ones, the recombinants. Always remember 3 plus 3, 6 are the new types, that is the non-parental types. They are not like the parents. They are going to be what? Round and green. They are going to be round and green and also wrinkled and yellow. New combination you have to make. Initially it was with green. Initially this was with yellow. Make a new combination. Works? Cool? Chalo. So basically, new parental combination 9 plus 3 plus 3 plus 1 is equal to what? 16. So out of 16, 3 plus 3, 6 are the new ones. So 6 by 16. Works? Children, do not go by the question, understand the concept. With that concept, you would be able to solve lots and lots of questions. And hi, hello. Yeah, a woman has hemophilic brother and normal parents. What is the possibility of her children having the disease? Hemophilic brother. But whenever you say a boy is hemophilic, this particular condition is known as a man being hemophilic, my dear children. And if a man has to be hemophilic, it has to get this particular gene from the mother, from the father. It is going to get only the Y chromosome, not the X chromosome. So mother has to be at least a carrier. Always remember that. In this condition, the mother is a carrier. Yeah. So woman has hemophilic brother. Okay, what about the woman itself? This is the brother. What about the woman? So if this is a condition, my dear children, I can say the woman can be XX, that is normal person, or she can be this type of person, that is a carrier. She can also be a carrier. Now, what is the possibility of our children having the disease if she marries a man who has no history of hemophilia? If a man is there, man cannot be a carrier, my dear children. If he is not having hemophilia, then it is XY. Now, I am going to cross her, that is a carrier female with a normal man. What do I get? This with this, you get a carrier female. Yeah, this plus this, I get a child who is having the disease, and this is a male child, my dear children. And now, what do you call it? This plus this, a female, normal female, and this plus this, a normal male. So what can I find out of this? I can find that there are 2 sons, and out of that, 1 son is having hemophilia. So can I say 50% of the son is hemophilic? Out of 2, 1 is hemophilic. So that means 50% of the sons are hemophilic, my dear children. And 50% of what? The daughter would be a carrier. But here that is not mentioned, so it is all cool. Works? Cool? Chalo. Next, the inheritance pattern of a gene over generation among humans is studied by pedigree analysis. In pedigree analysis, what do you study, my dear children? You talk about Mendelian trait. That means diseases that are going to be inherited. Okay, nothing else. Mendelian trait, you don't have to even read all the options out there. Just directly go with Mendelian trait. Okay, in pedigree chart. Now going ahead, pick out the correct statement. Hemophilia is a sex-linked recessive character. Yes, it is. Okay, hemophilia and color blindness, both of them are. Down syndrome is due to aneuploidy. What do you mean by aneuploidy? Addition or deletion of chromosome is known as aneuploidy. Okay, under aneuploidy you have 2 conditions, trisomy or monosomy. Trisomy means you are getting 1 chromosome. Okay, monosomy means you are losing 1. So in the case of Down syndrome, 21st chromosome, always remember, 21st chromosome is going to be additional chromosome. What are the features of Down syndrome? Remember that, broad pancreas, and then basically they would be mentally retarded. Please remember, if in the question it says that they are intelligent, it would be wrong. Okay, please go with that. And short stature, there will be furrowed tongue and lot of things are there. Okay, so 21st chromosome, an extra chromosome is there. This is known as trisomy. Okay, apart from this, aneuploidy conditions are what? Klinefelter syndrome and Turner syndrome. In the case of Klinefelter syndrome, you have XXY, 1 extra X chromosome is there. In the case of Turner syndrome, you have XO condition. That means initially it should have been a female, but you do not have any other chromosome up here, so it will be XO condition. This is Turner syndrome, this is Klinefelter syndrome. Works? Okay. Now, phenylketonuria is an autosomal dominant disorder. I told you in your NCERT there is nothing dominant mentioned in the paragraphs, only under diagram, and that is one myotonic dystrophy. This is wrong. Phenylketonuria is an autosomal recessive disorder. Correct. Sickle cell anemia is an X-linked recessive gene disorder, my dear children. Wrong. It is what? Autosomal recessive disorder. Works? Cool. Shall we go ahead? Yes. Select the correct basis of DNA, RNA, amino acid, beta gene. Okay. In the case of, I told you, in the case of mutation for sickle cell anemia, it was initially GAG, but it converts to GUG. How do you remember it? Ma'am, A comes before U. Okay, always remember. So A comes before U. Initially it was A, it became U. So initially it was coding for glutamic acid. Now it is coding for valine. Again, G comes before V. So please remember G and V first. Okay, first G and then it get coded by V. What type of protein is it affecting? It is affecting the beta globin chain. And also one more thing, sickle cell anemia and thalassemia, which one is qualitative disorder, which one is quantitative disorder? Quantitative disorder is thalassemia. Always remember, quanti, there is a T word and thalassemia also has T. Okay, qualitative is sickle cell anemia. Works? Cool? Chalo. Next, mother and father of a person with O blood group have A and B blood group. Okay. A person is having O blood group, mother and father are having A and B blood group, my dear children. What would be the genotype of mother and father? With this, you have to remember the whole thing up here. Okay, so if you say that the bacha is O, my dear children, then it's a recessive condition. Both the genes should be like this. O can be only expressed in this condition. So if that has to happen, my dear children, it should be getting the gene from here, from the mother, one gene from the mother and one gene from the father, then only it can happen. So basically, if she is A, then it has to be IA, and if he is B, it has to be IB, because A is dominant over O, B is also dominant over O. Now when these two genes are going to come together, it will be like this. So can I say mother is heterozygous for A? Yeah, heterozygous for A, and also the father would be heterozygous for B. Homozygous mean this is this condition. Are they homozygous? No, my dear children. So please go ahead with this. Works? Ma'am, please upload this PDF. PDF is already uploaded in the WhatsApp channel. Okay. Now, what I was saying, my name is Asmin Joy. Yes. Now, the result of the following experiment. So, basically, I'm just going ahead and telling you the experiment of Griffith. Okay, Griffith had pneumococcus bacteria, and there are two strains, R strain and S strain. R was the rough strain, S was the smooth strain. Smooth strain because it has an extra membrane out there. Okay, extra polysaccharide thing out there. Now, this was the virulent one. For example, if I am infected with S strain, I would die. Okay, it is the infectious one. So basically, he did the experiment where he took the rough strain, he put it in the mice, and he found that the mice lived. So the rough strain is not a problem. He took the smooth strain, put it in the mice, and the mice died. So S strain is a problem. Okay, then what he did, he heat killed the smooth strain. That means he treated with heat. And now then he found that the mouse was still living. How can that happen, my dear children? Probably certain changes were happening over here when they heat killed. Now one more thing was done. Rough strain was put, which was not causing any trouble, and it was mixed with heat killed, which was also not causing any trouble. Then it was put in the mice died. Why? Because probably from the heat killed one, something went to the rough strain. Yeah, that might have happened. Now, he got to understand this, but he couldn't tell what is genetic material. So Avery, MacLeod and McCarthy had to go ahead. And what did they do? They treated with something that is known as DNase. Always remember, it's not RNase, protease, nothing. So when they treated with DNase, okay, when they treated this particular combination with DNase, all the DNA went off, okay, it got cleaved off. So now do you think transformation can take place? No, transformation didn't take place. So they got to understand it's the DNA that is getting transferred. So in the question, if it is RNase, if it is protease, just understand it is wrong. It has to be DNase. I hope you got it. Works? Chalo, let's go ahead. Which of the following events would occur in the lac operon? My dear children, please understand the lac operon with me right now. So over here, you have three structural genes, Z, Y, and A. Zia, okay, you remember like that. Z codes for beta-galactosidase, Y codes for permease, and A codes for transacetylase. So what exactly happens is, there is a regulatory protein over here. This regulatory protein produces a protein that is known as repressor. And what does this repressor do? It goes and binds on the operator region. Remember, it's operator, no other region. It will go and bind on the operator region. Just before the operator region, what do you have? You have the promoter region. And this is where the RNA polymerase will come and bind for doing the transcription. But can it move ahead? No, because this particular thing is there on the operator region. Now what has to come to make it switch on? This is switched off condition. For transcription to happen, lactose has to get inside. So lactose over here will be known as an inducer. It can be lactose or it can be allolactose as well. It comes in, binds with the repressor. Now the repressor is not free to go and bind over there, which is why transcription will happen and beta-galactosidase, permease and transacetyl is formed. ZYA. And what is the function of beta-galactosidase? Basically, it breaks down lactose into two things, glucose and galactose. Works? So this is how it works, and when it breaks down, the E. coli gets the energy. What is the role of permease? It increases the permeability such that lactose can get inside. Works? Chalo? Yes. So the inducer molecule binds to the repressor and RNA polymerase binds to the promoter sequence. That will be the thing you can read. Now, out of 23 pairs of chromosomes in male, first and the 46th chromosome respectively possess how many genes? That means you're talking about the first chromosome, and what is the 46th chromosome? It is a Y chromosome, the smallest chromosome. So basically the number of genes on the first chromosome is the highest, and that is 2968. And in the case of Y chromosome, it is just 231. Works? Okay. Next. Read the following statement and choose the correct option. Nitrogenous base is linked to the pentose sugar through N-glycoside linkage. My dear children, this is the pentose sugar, 5-carbon sugar, and it is linked to the nitrogen base with the help of a bond. This bond is known as N-glycosidic linkage. This is absolutely correct. Now, phosphate group is linked to the 5-OH of nucleotide through phosphoester linkage. Remember, over here, on the 5th carbon, this is the 5th carbon, it is linked to a phosphate, and this bond is known as phosphoester linkage, not diester. When does it become diester? When this nucleotide bonds with another nucleotide. Okay, so when you say that basically in one nucleotide you are attaching the 3rd carbon, in the other nucleotide you are attaching the 5th carbon. You're attaching the 5th carbon. So basically there's a bond over here, there's a bond over here. So phosphoester bond becomes what? Phosphodiester. That's the only time you say phosphodiester. If you're just talking about one nucleotide, it has to be just phosphoester. Remember. So this is also correct. Now 2 nucleosides are linked through 3,5 N-glucosidic linkage. Wrong. What do you mean by nucleoside? You remove this phosphate, the thing that is remaining over here, pentose and the nitrogen base is known as nucleoside. Now, negatively charged DNA is wrapped around positively charged histone octamer. Do you think DNA is negatively charged? Yes, DNA is negatively charged, always remember that, and it is wrapped around histone octamer. Histone octamer is positively charged. Why is it positively charged? Because it has 2 amino acid, lysine and arginine. Okay, lysine and arginine, which is why it is positively charged, onto which the DNA would be wrapping around twice. And together, this forms a structure which is known as nucleosome. What do you call this? Nucleosome, my dear children. Now, chromatin that is more densely packed and stains dark is known as euchromatin. Wrong. My dear children, you have 2 types of chromatin. Okay, basically the DNA is pretty long. You have to compact it. So you coil it around the nucleosomes. Yeah, and this is how it looks. This is how it looks. So it can be loose or it can be tight. The tight one is known as heterochromatin, okay? And the loose one is known as euchromatin. So which one will be active transcriptionally? That means RNA polymerase can come and bind to which one? Basically this one, because it is loose, it has space, it will do transcription. So always remember, euchromatin is transcriptionally active. Heterochromatin is not transcriptionally active. It stains really dark because it is too compact. It is going to stain really less because it is too loose. Works? Got it? Chalo. Next one. Which of the following radioisotopes is not suitable for DNA labeling? My dear children, in DNA, what all do you find? You have phosphate, you have nitrogen, you have hydrogen, everything. Do you have sulfur in phosphate? No. So if this in the option you have sulfur, that means it is the wrong one. In the case of proteins, you have it. For radio labeling proteins, you need a sulfur, but not in the case of DNA, so sulfur would be out. Works? Next thing. A monocystonic RNA, mRNA, can produce several types of polypeptide chains. Always remember, you to have 2 types of things, monocystonic RNA and polycystonic RNA. What do you mean by monocystonic RNA? Basically, this RNA would be coding for just one gene. Okay, it will be having just one gene coding for a single protein. Polycystonic, just like E. coli or any bacteria, my dear children, they will be having multiple genes in one particular unit. Okay, you saw ZYA, three genes. Yeah, three things are being coded. So that is polycystonic, and that you find in prokaryotes. So always remember, P for polycystonic and P for prokaryotes, whereas monocystonic is found in eukaryotes. Yeah. Now, terminator codon is present on the mRNA. Do you think terminator codon is present on the mRNA? Yes, my dear children. What are the 3 terminator codons that you have? You have 3 of them: UA, UGA, and UAG. I'll tell you: UA, UGA, and UAG. Okay, works. These are the 3 terminator codons. Repeat with me: UA, UGA, and UAG. What is the start codon? You have just one start codon, and that is AUG, and AUG also codes for what? Methionine. It also codes for methionine. Works? Next thing, E. coli cells with mutated Z gene. Okay, suppose in the lac operon, Z gene is mutated. What will not happen? Z is for beta-galactosidase. Hence, my dear children, beta-galactosidase would not be produced. Always remember that. Y for permease, A for transacetylase. Going ahead, find the mismatch. Okay, RNA polymerase 1, 2, and 3. We have 3 types of RNA polymerase in the case of eukaryotes, and 3 of them are responsible for transcribing different things. Okay, what one is the first one transcribing for? Always remember, first priority is given to ribosomal proteins. Okay, so it is rRNA. So RNA polymerase 1 will be transcribing rRNA. Ribosome is made up of rRNA and ribosomal proteins, my dear children, and that is the first priority. So R and R first priority. Okay. Now RNA polymerase 2, what does it transcribe? It transcribes the H in RNA. hnRNA. You know, from the DNA, you get hnRNA. This hnRNA will be forming the mRNA. So it's the hnRNA that is getting transcribed first. Okay, now RNA polymerase III transcribes tRNA, my dear children. The last priority is what? tRNA, 5s rRNA and snRNA. If we cannot remember this, at least remember this. First priority rRNA, second priority hnRNA or mRNA, third priority T for third and T for tRNA. Works? Cool? Children, don't get worried if I'm going really fast. It's because I want to save much of your time and want to explain a lot of things. Okay, so please don't be in that sense. Histone octamer differs from nucleosome in lacking what? You know, talk about histone octamer, that means you're talking about histone protein. 1, 2, 3, 4, 5, 6, 7, 8. How many histones? 8 histones are there. How do they differ from the nucleosome? Nucleosome is basically when on this particular histone octamer you wrap what? The DNA, and DNA is what? Negatively charged. So negatively charged DNA. Okay, how do they differ? It is because in the nucleosome you also have what? The DNA along with it. That's all. Next thing, in genetic coding, triplet codons make 64 possible codons. Okay, basically we have 4 nucleotides, what do you call A, G, C and T. Okay, in which combination do they have to come to give 20 amino acids? If we do this 4 square, if they come in this, that is in doublet format, it could give you 16 codons. Is that enough for coding 20 amino acids? No, that won't be enough because repetition would be happening. So always remember, if it is doublet, it will be coding for 16. Okay, if it is like this, it will be coding for 64, and this is what exactly is happening. Now out of this, how many of them are effective ones? Can you tell me how many of them are effective ones? Only 61 of them are going to effectively code for amino acids. 3 of them are going to be stop codons. Works? Chalo. Next thing. Find the mismatch. You have bacteriophage 5386 nucleotides, correct? Bacteriophage lambda 58502, correct? E. coli 4.6 in 10 to the power 6 base pairs, correct? My dear children, they are just making you confused by giving big terms. It's just for you to identify this. The easiest one. Haploid human DNA. Can it contain 6.6 in 10 to the power 9 base pairs? Wrong. Haploid, half of it. That means it will be 3.3 into 10 to the power 9. This is for diploid, 6.6 into 10 to the power 9. Okay? Works? Works? Chalo. Next. If the DNA of mammalian cells contains 6.6 into 10 to the power 9 base pair, the length of the DNA would be, if, my dear children, we have 6.6 into 10 to the power 9 base pairs, what is the space between each base pair in the DNA? It is 0.34 nanometers. 0.34 nanometer. Convert into meter and you multiply. You get 2.2 meters. What is it? 2.2 meters. Okay? Works? Next. DNA differs from RNA. How? Deoxyribose sugar instead of ribose. Yes, in the case of RNA you have ribose sugar. Here you have deoxy. In the case of DNA you have thiamine. In the case of in the case of RNA you have uracil. Okay, and always remember, thiamine came from uracil, so it is known as 5-methyl uracil also. Thiamine is also known as 5-methyl uracil. Okay, next, H instead of OH. So if you talk about the pentose sugar which is there, my dear children, in the case of RNA, here you have OH on the second position. But in the case of DNA, you just have H. You just have X. That is why H. It is known as deoxy. Oxygen is lost. So all of them are going to be correct. Okay. Next one. Next chapter. Okay. So after how many divisions does functional megaspore result in mature embryo sac, my dear children? Functional megaspore. You have one functional megaspore. Okay, you got 4 of them. 3 of them degenerated. 1 of them is left. And basically, this one of them have to go how many divisions? Basically, 3 mitotic divisions. If you talk about a functional megaspore, it will undergo first mitotic division, giving rise to 2 nuclei. Remember, the nuclei is not surrounded by cell wall suddenly. Okay, it will after some time only it will happen. So again, this will divide. You get 4 more. Again, you will divide. You get 8 nuclei at the end. Inside one functional megaspore, you get what? 8 nuclei. Or basically inside one embryo sac, that is a female gametophyte, you get 8 nuclei, and the 8 nuclei are arranged in which order? This order, my dear children. You have 2 nuclei over here. These cells are known as synergids, and the top of it you have one X cell. Yeah. On the top of it, at the topmost end, you have antipodals, 3 antipodals, and in the between you have 2 nuclei. That is known as 2 polar nuclei, and this belongs to a biggest cell that is known as central cell. Always remember, if you come out the number of cells, 1, 2, 3, 4, 5, 6 and 7. 7 cells are there, but how many nucleus is there? 1, 2, 3, 4, 5, 6, 7, 8 nucleus is there. So it is 7-celled, 8 nucleate condition. Okay, always remember that. And what I had to tell you, in the case of synergids, they have something like this that is known as filiform apparatus. What is the use of filiform apparatus? It is to guide the pollen tube inside the synergids. Remember that. It is to guide what? Pollen tubes inside the synergids. Works? Okay. Now, what do I have to tell you? Okay, the ploidy over here. What are the ploidy of all the cells over here? So, if you talk about antipodals, all of them are haploid. N. Okay, if we talk about synergids, they are also haploid. If we talk about XL, that is also haploid. But what about central cells? Since they have 2 nuclei and each of the nuclei, my dear children, have NN. They are both haploid. So can I say in total it will be 2N? Yes. So the ploidy of central cell will be 2N, the only different cell. Okay. And always remember, micropolar end is the place where you would be having the XL. Okay. And antipodal cells will be always at the chelazal end, nowhere else. Fine. Works. I'll be talking about pollen grains, okay? Now, in Commelina, flowers are produced on. So, my dear children, Commelina, oxalis and viola are examples of what? Are examples of clistogamous and chasmoglamous flowers. That means open flowers and closed flowers. Okay? In that, you find leaves, flowers at 2 positions, at the top and also in the underground root or rhizomes, my dear children. Okay? Now, one very important thing over here. If we talk about the closed ones, what do you think will be happening? Always autogamy is going to happen, nothing else. Nothing else is going to happen in the lower flowers. In the upper flowers, my dear children, you can have autogamy, you can have geitonogamy, that means flower to flower of the same plant, or it can be xenogamy, that means from the different plant also it can come over here. Okay, so 3 examples, Commelina, oxalis and viola. Works? Chalo. Now, next. Okay. Tapetum nourishes the developing pollen grains. So, in the case of, my dear children, if you talk about microsporangias, the structure, okay, remember, listen over here, you have sporogenous tissue. Outside the sporogenous tissue, you have tapetum. Tapetum is the one that nourishes the sporogenous tissue, the cells of the sporogenous tissue, that is the future pollen grains. And inside the tapetum, the nucleus is not one, it is not uninucleate, it is binucleate, okay, it is binucleate because karyokinesis happened. The nucleus divided, but the cell didn't divide. Always remember that. Now, outside it, you have 3 layers. One is middle layer, the outer, then is endothecium, and then is epidermis. Okay, epidermis, endothecium, middle layer. 3 of them are responsible for dehiscence of anther, for breaking down of the anther. Okay, now inside that, the sporogenous tissue, each of the cells which are there, they are going to act like a microspore, mother cell. They are going to undergo meiosis. At the end, you are going to get 4 microspores, and 4 of them are going to form pollen grains. How does a pollen grain look? Listen to me carefully. You have the pollen grain. This is the plasma membrane. Outside that, you have one layer that is known as intine, and intine is made up of 2 things, cellulose and pectin. Outside that, you have one more layer that is not continuous, and it is known as what? Just a second. Let me breathe. It is known as exine, and exine is made up of what? Sporopollen, the most resistant material out there on earth. Okay, it can resist everything. Now, you see the openings, these are known as germ pores, and through the germ pores, my dear children, what is going to go? The pollen tube is going to be arising. Where do you get the pollen tube from? The intine itself. Everything forms the pollen tube. Now, my dear children, inside the pollen grain, you have 2 types of cell. One is generative cell, and the other one is vegetative cell. Vegetative cell is the biggest cell. Inside the cytoplasm of that, you have a small little generative cell. G for generative and G for gametes. Generative cells are the ones that are going to divide and form 2 gametes later on. Okay, they are going to form 2 gametes later on. So basically, for us, in the pollen tube, what all is there? Basically, you would be having one tube nucleus. Tube nucleus belongs to the vegetative cell. Okay, and you would be having 2 male gametes. Now listen to me. 60% of the angiosperms are going to go in this particular stage where they just have 2 cells. Okay, 40% of the angiosperms are going to go in 3 cell stage. 3 cell stage. So remember, 4 and 3 are close. So you can remember like that. Works? Okay. Oh, you wait. Now, hilum represents the junction between ovule and the funicle. Very important. Listen to me carefully. So, if you talk about the ovary, which is over here, okay, you have the ovule. This is the placenta. Placenta is attached to the ovule with the help of a structure that is known as funicle. And the funicle over here attaches on the area that is known as hilum. So, hilum, my dear children, after some time, it will be like a scar on the seed. So, what is the scar? Scar represents hilum. And what is the pore? Pore represents micropyle. Okay, always remember that. Works. Now, in aquatic plants like water hyacinth and water lily, pollination is by water. Wrong. Water hyacinth and water lily do not do pollination by water. It can be wind, it can be anything, but not water. Vallisneria and all the other things would be doing by water. Okay. Now, the primary endosperm nucleus is triploid. Do you think it is triploid? Primary endosperm nucleus, how do you get? My dear children, listen to me carefully. This is double fertilization. What happens over here? One male gamete comes and fuses with the egg cell, okay? And this is going to form what? One male gamete comes and fuses with the egg cell, and this is going to form zygote. Yeah? Now the second male gamete which is there, it comes and fuses with the two polar nuclei of the central cell. So the male gamete is also carrying an N. So N, it becomes triploid, that is 3N. What is the product? What do you call this? Primary endosperm nucleus. Works? It is known as primary endosperm nucleus. Fine, children. Oh, I hope you are studying, right? Now, basically, tell this answer. Oh, come on. Yes. The male gametes of the rice plant have 12 chromosomes. Male gamete, it is having 12 chromosomes, okay? The chromosome number in the female gamete will also have 12 chromosomes. What happens in the zygote? 12 plus 12, 24, yeah? And what about the cells of the seedling, my dear children? All the cells which will be formed from the zygote will be having 24 only. So can I say 12, 24, 24? I can say that, so this becomes your answer. Okay, next question, very important question. The 2000-year-old seed excavated from King Herod's place at the Dead Sea belongs to what? Phoenix dactylifera. It is known as Phoenix dactylifera, my dear children, 2000 years old. Now in the question, if it is 10,000 years old, the answer would be Lupinus articus. Always remember, 2000 it is for Phoenix dactylifera, 10,000 it is for Lupine articus. Okay, works? Will you be able to remember that? Yeah? Children, we'll talk about RCB later. Yes, RCB will win, lose, we'll see that later, okay? Now let's see if you can win or lose. Now 100 functional megaspore gives rise to what? 100 functional, 1 megaspore gives rise to 1 embryo sac. Embryo sac is that female gametophyte where the female gamete is there, right? Now 100 of them would be giving rise to 100 embryo sac as well. Again, let's go ahead with the answer. Autogamy. Autogamy can occur in chasmogamous flowers if autogamy means what? Self-pollination. Pollen grains from the same flower on the stigma of the same flower. When do you think this can actually happen? Pollen matures before maturity of the ovule? No, there is no synchronization. Here also no synchronization. Both pollen and ovules mature simultaneously. There is synchronization, and that's when autogamy can happen in an open flower, that is chasmogamous flower. Always remember, clistogamous are closed ones. CL for closed, CL for clistogamous. Works. Now in the embryos of a typical structure enclosing reef Promodia, single cotyledon and respectively, you're talking about monocotyledons. Monocotyledons, name suggests mono, that means only one cotyledon is there. What do you call this cotyledon? You call this cotyledon as scutellum. Okay, and basically if you talk about the structure, it looks like this. This is the embryonal axis. From here you get the cotyledon. Yeah, this is going to form the plumule. This is going to form the radicle. Below the radicle you have root cap. Covering the root cap you have something else which is not found in dicots, and that is known as coleorhiza. Coleorhiza. Always remember, R for rhiza and R is found in the root only. Here, leaf Promodia. Okay, at the top of the leaf Promodia, what do you have? Coleoptile. Coleoptile. P for plumule, P for coleoptile. You can remember like that. Not P for coleoptile, but yeah, P is there. Right? So this is a scutellum, and since they are monocots, they also have endosperm still persisting. They would be having endosperm which is still persisting. Fine. So your answer would be coleoptile and scutellum. Works. Next thing. Find the mismatch. Fall fruits other than the ovary, thalamus taking place in the fruit formation. Okay, false fruits are the ones where the not just the ovary but anything else also can help in the formation of fruits. That is correct. Example is cashew nut. Example is your example is what do you call this? Apple. Okay, in the apple basically the thalamus is participating in the formation of fruit. Now parthenocarpic fruit, fruits developed without fertilization. Remember, there are two terms, you have to remember it properly. Okay, that is what basically when suppose this is ovary, this is the ovule, yeah? When the ovary without fertilization is forming the fruit, what do you call this? Ovary without fertilization forming the fruit, you call this parthenocarpic fruit. Why does this happen, ma'am? Basically, there are certain hormones that plays a major role over here. And in this case, since fertilization has not happened, the ovule is not transforming into a seed, and hence, these parthenocarpic fruits would be seedless. Example is your banana, okay? Talking about apomixis seed, what happens over there? Here, in the case of ovule, fertilization is happening without fertilization, you are going to form seed. Without fertilization, when you are going to form seed, it is known as apomixis. Okay, where does it take place? In the case of grass and then things like that. And along with apomixis, there is one more thing, that is polyembryony. Polyembryony means multiple embryos are being formed inside the ovule, my dear children. From where? Probably from the cells of the nucleus which surrounds it. So they get inside, they form this. Where do you see this? This is found in citrus plants. Okay, so apomixis is basically found in grass and Asteraceae family and things like that. Polyembryony, if you are asked for a specific example, I'll tell you it is citrus fruits. Okay, orange and everything that comes along. Now, true fruit is when fruit develops from the ovary itself. Correct. From the fertilized ovary. Seeds, do you think they develop from the ovary? No, seeds come from the ovule, my dear children. Fertilized ovule is known as seed. But in the case of upper mixes, that is not the case. Next, the correct chronological sequence of the next chapter, okay? Okay, first thing you have gametogenesis, gamete forms, yeah? And then insemination where the gametes are transferred, fertilization, okay? And then after fertilization, the zygote divides, okay? You get the embryo gets transplanted, and this is known as implantation. Okay, where do they get implanted, my dear children? In the endometrium. In which stage do they get implanted? It is known as blastocyst stage where they get implanted. Okay, and gestation where basically the baby is going to form, and parturition where the baby is going to be moving out. Works? Next. Read the following statement about seminal plasma. Okay, seminal plasma is made up of three things, secretion of three things: seminal vesicle, prostate, and bulbourethral. Usually children confuse with this and this. No, it's three of them together, you call it seminal plasma. And now listen to me carefully. Seminal vesicle releases something that is known as sucrose. It is known as what? Sucrose. Wait, not sucrose. Wait. Don't learn that, okay? So seminal vesicle produces something that is rich on fructose, and fructose is that something that provides nourishment to these sperms, okay? Now talking about prostate glands, prostate releases two things, that is calcium and also certain enzymes. So in total, what all did you get? You got fructose, you got calcium, you got enzymes, okay? You got fructose, you got calcium, and also you got what? Enzymes. So this is what you get in seminal plasma. And also remember, bulbourethral gland is there for lubrication. So can I say it is rich in sucrose? No, it is rich in fructose and calcium, and it contains enzymes also. That is all correct. Okay, works? Now going ahead, it came over here. The ploidy of spermatogonia, primary spermatocyte, secondary spermatocyte, and spermatid is what? So basically over here, if you see, you have spermatogonia, my dear children. Some of it, they differentiate, okay? They differentiate and form what? The primary spermatocyte. The primary spermatocyte will be having 2N. It has not yet started dividing, yeah? The primary spermatocyte will be dividing into two. You get what? 2 secondary spermatocyte. That means here meiosis 1 is completed, and when meiosis 1 is completed, you get N, my dear children. Haploid, it becomes haploid. Now the secondary spermatocyte again divides into 2, here also divides into 2. What do you call this? You call this as spermatid. It's like a kid, it is not mature, yeah? To make it mature, you have to convert into what? Spermatozoa. From here to here, all of them are going to be N. So you already got your answer. This is going to be 2N, this is going to be N, and this is going to be what? N as well. Now listen to me. Spermatid to spermatozoa conversion is known as what? Spermiogenesis. Spermiogenesis. And over here, there is a major role of Sertoli cells. Sertoli cells. Okay, always remember that. So Sertoli cells release some factors which is very important for spermiogenesis. Works? Okay? Now, going ahead. Okay, the first meiotic division during oogenesis is completed at the stage of. First meiotic division gets completed at the stage of primary oocyte. They divide into secondary oocyte, my dear children. Within what? The tertiary follicle. Listen to me, I'm going to talk about oogenesis over here. Similarly, oogonia is there. Yeah, oogonia are formed in the, embryonical stage only. Yeah, and this oogonia, basically one of them, they differentiate into what? Primary oocyte. The primary oocyte is going to undergo meiotic division. This is what? 2N stage. Yeah, it is undergoing meiotic division. One small cell, one big cell. Unequal division. This is known as secondary oocyte. This is known as secondary oocyte. How? What is the stage? N, haploid. It had become haploid now. Okay. So if you are asked about ploidy of primary oocyte, it is diploid. It has 6.6 into 10 to the power 9 base pairs. Always remember that. Now it also does meiosis, right? Again you get N. This is what? Ovum, my dear children. This is what? Ovum. So first meiotic division gets completed in the tertiary follicle. First meiotic division gets completed in the tertiary follicle. Then you have secondary oocyte. Secondary oocyte remains in the tertiary follicle. Basically what happens? The tertiary follicle matures into graphene follicle. So basically in the graphene follicle, you will be seeing what? The secondary oocyte itself, it has not completed its division. When does it complete its division? After fertilization happens, that's when it completes its division. Always remember that. And in the case of graphene follicle, the secondary oocyte is going to release something in the outer layer that is known as zona pellucida. And outside zona pellucida, they have something else, that is corona radiata, that is not made by secondary oocyte. Always remember only one. Okay? Works? Now, next thing. Which set of hormones are secreted during pregnancy only? Only during pregnancy, my dear children. It's not estrogen. Estrogen can be released anytime in our body, even progesterone. Okay? So, whenever you have these options, cut it out. Even cortisol can be released anytime in our body. What is it? HCG, HPL and relaxin. Okay? listen to me. There are 4 placental hormones, that is HCG, human chorionic gonadotropin, and then HPL, and then estrogen, and then progesterone. But these things are usually being released in our body also. But placenta, if you talk about placenta, if you're asked about placenta, they release these 4 hormones. Now, apart from that, in the case of our ovary, during later phase of pregnancy, it releases something that is known as relaxin, so that the muscles are relaxed and baby can be moving out very easily. So, only hormones that are released during the pregnancy is HCH, HPL, and relaxin. Okay, works? What's the time? Yes. Now, embryo at 8 to 16 cell stage is known as what? Embryo at 8 to 16 cell stage, it is known as morula. It is known as what? Morula, my dear children. Morula will again keep on dividing, form the blastocyst. And remember, in the blastocyst, you have 2 types of cells. The outer layer of cells is known as trophoblast. The inner cell mass is known as, inner layer of cells is known as inner cell mass. This inner cell mass is something that is going to actually form the baby. Yeah? What happens to trophoblast? Trophoblast is there, responsible for the formation of chorionic villi, and then later placenta and lot of things. Okay? Always remember that. Yes. Next, the fetal ejection reflex in humans triggers the release of. Listen to me carefully about the baby. Okay. So what happens, baby? Say, what happens actually? Oh my God. Okay. So what actually happens is that suppose there is a baby, fully developed fetus. Okay. This fetus tries to move out. When it tries to move out, there will be certain stress receptors on the cervix that becomes activated. Now, these stress receptors sends neuronal signal to what? Mother's pituitary gland. Which pituitary, my dear children? Posterior pituitary. Not anterior, posterior pituitary. And then the posterior pituitary will be releasing what? Oxytocin, which is a hormone. Okay, now what else is happening in the womb, my dear children? The baby tries to pushing out, that is fine. The placenta is also releasing something that is known as estrogen. The placenta releases estrogen, and this estrogen basically is going to make all the receptors, oxytocin receptors on the myometrium, the muscular layer, active. Now what happens? The oxytocin which is released from the mother comes over here, binds over here, and there is what? Contraction, repeated contraction, which is why the baby would be moving out. This is what happens. So in fetal ejection reflex, what happened? Oxytocin from the maternal pituitary is going to be released. If you have any more options apart from this, you can go ahead with what I try to explain. Okay? Works? First year questions, I would be coming to that. Now, in which period during embryonic development heart starts beating approximately 60 times per minute? My dear children, listen to me carefully, okay? There are few months mentioned in your NCERT, you have to remember that exactly. Okay, first month, heartbeat. First month, heartbeat. So first month is made up of 4 weeks. So after 4 weeks, you will get heartbeat. Okay. Second month, you will be having limbs and you will be having digits and also external genitalia, also organ system and things like that. Okay. What happens in the second month? After second month, you will see all these things: limbs, digits, external genitalia, and the organ system. Now directly skipping to the 5th month. Okay, 5th month, my dear children. What happens in the 5th month? Basically, in the 5th month, the baby says, I want hair on my head and I also want to move around. So hair on the head and also moving around. Movement, first movement is found in the 5th month. Now in the 6th month, last month given in your NCERT, what happens? Eyelids separate, eyelashes are formed, and also there will be hair on the body. So there are 4 months mentioned in your NCERT. You should be knowing according to trimester wise also. So my dear children, if you asked what happens in the first trimester, so first trimester means you're talking about the first month, second month, and third month. Third month is not given in NCERT until second month is given. So you have to tell heartbeat is formed, ma'am, and basically limbs and digits, external genitalia, organ system, everything is formed. Okay, we talked about the second trimester. Second trimester means second 3 months, and after that 1 more 3 months. 3 plus 3, 6. Okay, that is second trimester. So after 6 months, what happens? All these things that you told, all of that would be happening. Works? Now, 50 secondary oocyte in female and 50 secondary spermatocyte in males gives rise to what? 50 secondary oocyte, 1 secondary oocyte can give rise to only 1 ovum because it is an unequal division. Remember that? So it will be, if this is 50, this will also be 50. But in the case of spermatocyte, this is not like that. 1 spermatocyte can give rise to 2 spermatids, right? So 50 into 2 is basically 100, so 100 sperms. Works? Ma'am ne paper kharid liya, nahi kharida. Okay, next. The presence or absence of hymen is not a reliable indicator of virginity or sexual experience. It is absolutely correct. Presence or absence of hymen is not a reliable indicator. This is correct. The sex of the fetus is determined by the father and not the mother. That is also correct because fathers are heterogametic. X chromosome, Y chromosome. Either of them can be coming together. Works? Now, next question. Okay, which of the following cannot be detected by a developing fetus by amniocentesis? Amniocentesis is basically when you are going to take the amniotic fluid around the baby. This would be having the cells of the baby which got shut down. So basically, you are going to talk about the chromosomes, the genes and everything. So any genetic disorders can be found out. Okay, people use it for finding the sex of the child. That is wrongdoing. Okay, but jaundice is not a genetic disorder. This is not something that you can find with the help of amniocentesis. You can find Down syndrome, Klinefelter syndrome and all the other things. Works. Next thing, given below are the 4 methods. You should know what is it. Okay, so copper T, my dear children, copper T is basically the one that is going to hold the sperms, okay, hold the sperms and not allow it to move. So basically it does not allow movement and also reduces the fertilizing capacity of the sperms. Copper T is a what? It is copper releasing IUD, okay. Now vasectomy means you are cutting the vas deferens. If you are cutting the vas deferens, you are blocking the gamete transport. Condom again is a physical barrier, does not allow the sperm and the egg to meet. Okay, now pills. What about pills, my dear children? Pills are going to prevent ovulation. Why? Because in pills you have progesterone and estrogen combination, and in normal level they are going to help in ovulation and they are going to help in maintaining endometrium, but in abnormal level they are going to do the opposite. No ovulation properly, endometrium becomes very thin and the cervix become very thick because of the mucus plug. Okay, so sperm cannot enter. Always remember, whenever you're talking about hormone releasing IUDs, the pills, everything, you have the same effect. Okay, there's only one pill which does not have any steroids being produced, and that is Saheli, produced by CDRI, Central Drug Research Institute, Lucknow. Okay, that is the only non-steroidal pill that you have. It's a once in a week pill. Okay, CDRI starts, I told you, yeah. And what is this? This is condom. This is what? Implant. Implant, you put it on the skin and they release the hormones. What is this? This is copper T, and this is what? The female condom which is there. Works. Now, administration of progesterone, estrogen combination of IUD are effective within how many hours of coitus? 72 hours of coitus you have to put it, otherwise it won't be working. Working? Cool. Next thing. Very important. Medical termination of pregnancy is considered safe up to how many months? My dear children, for how many after... Okay, how many weeks can you do MTP, medical termination of pregnancy? Almost 24 weeks you can do, but up to 12 weeks it is considered to be safe. Always remember, up to 24 weeks you can do, but up to 12 weeks it is considered to be what? Safe. Remember that. Now, what are the factors in saying that reproductive health of the society is good? My dear children, there should be less maternal mortality rate. Mothers would be dying less. Mothers should be dying less, right? Second thing, the baby should be dying less, yeah? And there should be better postnatal care and better detection of cure of disease and all the things. My dear children, one very important thing, it can be coming in the option that basically as an initiative of government, they gave, one child, one norm and things like that. And it was more accepted in rural areas. Wrong. It is not rural areas, it is urban areas where it was accepted. So please go to the terms also. Okay, next thing, TI plasmid is useful in biotechnology and principles. Okay, TI plasmid, by dear children, it is there in agrobacterial tumescence. And what does this TI plasmid do? TI plasmid has an area, this is known as transfer DNA. What do you call this? Transfer DNA. This transfer DNA, when inserted into the plant would be having tumor. Can we use the same mechanism for transferring what we need? Yes. So which is why what was happening is that we remove the tDNA from tumor-inducing plasmid, my dear children, and we put our genes, and the same agrobacterium went and infected the plant, and we got our thing put inside the plant. Okay, so in the case of plants, you always use agrobacterial tumescence. In the case of animals, you are going to use retroviruses. Always remember, HIV is a great thing for our body, right, for infecting our body. Same mechanism we are going to use. We are going to use disarmed retroviruses for putting things inside the human body. Okay, that is animals, not the human body, not just the human body, but also animals. Now this thing, very, very important, PBR 322. Okay, I'm not going to tell about this. I'm going to talk about, I'll make and show you. Listen, always over here you will be having origin of replication. What? Origin of replication. From here it will be starting. Okay. And then here you have ROP site. That is a protein needed for replication. That is ROP site. Now on both the sites, my dear children, you have resistance gene. You have two types of resistance gene. One is ampicillin resistant gene and this is tetracycline resistant gene. Okay, you have two types of resistant gene. Now, apart from that, you would be having restriction sites for certain enzymes. Two here, two here. So here you have BAMH1. Here you have SAL1. Suppose it says that basically I am going to cut down with the help of BAMH1. If a person is going to cut down with the help of BAMH1, my dear children, basically it is going to make a cleave over here. Do you think it will be tetracycline resistant anymore? It won't be tetracycline resistant anymore. So that is how question could be coming. So BAMH1, SAL1 over here, and here you have PVU1 and PST1. PVU1 and PST1. Over here in the ROP site also you have one, and that is the friend of it, that is PVU2. PVU2. So 2 over here, 2 over here, 1 over here, and 3 over here. Here you have EcoR1. Here you have clavin and also Hind3. Also what? Hind3, not 2, Hind3. Works? Krishna says, relax ma'am, we'll do. Yes, I know, I'm pretty sure you will do great in biology examination. Chalo, but anyway, I have to continue, yeah? So now, a plasmid has 2 antibiotic resistant genes, one for ampicillin and another for tetracycline. It is treated with restriction enzyme that cuts in the middle of the ampicillin gene. Okay, or it can be said that PVU1 is cutting. So you should know it is cutting in the ampicillin gene. Now do you think it can grow in ampicillin medium, my dear children? No, it is not resistant anymore. It won't be able to grow in ampicillin, but it can grow in tetracycline. Next thing, very important, which restriction enzyme can be used to cut this given gene? So over here it is mentioned, the sequence is mentioned, GAATTC and 5-GAATTC3 is for what? EcoR1. EcoR1 can make a cut over here. Always remember 5-GAATTC3. That is the polarity, 5 to 3 polarity. Okay. And Hind2, you would not be asked the base pairs, but you would be asked the number of base pairs. And that is what? 6. Just like this also, they also have 6 base pairs restriction site. Works? Okay. Next thing, which of the following steps is not involved? Okay, I'll just tell you what are the steps involved for making a cell competent. Okay, that means capable of taking in certain things. First one, okay, first one is basically treating the cell with divalent cations. Okay, divalent cations. Why do you need to deal it with that? Basically, you want to make the DNA which is out there neutral. Okay, DNA is negatively charged, and negatively charged cannot be passing down from the cell membrane inside the cell with the host cell, right? So basically, you have to treat it with divalent cations. After you do that, you are going to put it inside the ice. What all are you going to put it? The host cell where you want to transfer things and the thing that you want to transfer. Both of them you are going to put in the ice. Why? Because in the ice, my dear children, everything gets changed. Now, from the ice, you are going to transfer it into heat shock. Okay, you are going to give heat shock. This heat shock is going to give transient pores. There will be small pores through which the DNA can enter. And again, now you are going to put in ice. So first with divalent cations, then with ice, then with heat shock, then again with ice. Done. Okay, this is how you make the cell competent. And so which statement is incorrect? Retrovirus have been disarmed and are now used to deliver desirable genes into animal cells. Animal cells, then it is correct. Downstream processing, one of the steps of our DNA technology. Yes, downstream processing means you are purifying, you are filtering and everything. Okay, the important thing. So that is there. DNA is negatively charged, I told you. The presence of chromogenic substance give blue color colonies. There is blue-white colonies, my dear children. If you know blue-white colonies, it makes sense, right? If you do not know it, I'll tell you. So listen to me. Again, suppose this is a plasmid. Here I have a gene that is LAG-Z gene. LAG-Z gene codes for beta-galactosidase, right? So normal case it will code for beta-galactosidase, and suppose I am putting a substrate that is X-gal. It will react with the substrate and produce what? Blue color colonies. So usual condition is like this. But now suppose I am putting my gene of interest. Do you think it will be producing LAG-Z? It can produce beta-galactosidase now? No. So do you think the color change will be happening even if I put substrate? No. It will be giving me white colonies. So that is what is exactly asked over here. That is correct also. Now, next question. Gel electrophoresis, my dear children. Gel electrophoresis and elution are two important processes. Yes. What do you mean by elution? For example, in the case of gel electrophoresis, my dear children, first thing, listen to me carefully. Okay, in the case of gel electrophoresis, from where to where is the DNA going to move? From the cathode. Cathode is what? Cathode is negative and anode is positive. Cations are positive, anions are negative. But here it's ulta. Cathode is going to be negative and anode is going to be positive. And DNA is what? Negatively charged. So it is going to move from this direction to this direction. From to positive and not, because the normal one, the similar one would be repelling, right? So it will be moving over here in the anode. Works? Now, what else is happening? The smaller fragments are going to move fast. Always remember that also. Now what I have to say, if you want to see the DNA, okay, what you have to do? You have to treat it with ethidium bromide. What? Ethidium bromide, my dear children. And still you won't be able to see unless and until you put UV light. When you put UV light, what will happen? It appears as a bright orange color thing, bands. Okay, bright orange bands that you would be able to see. Now when I'm wanting to remove this DNA out of this gel, what do you call this process as? This is known as elution. This is known as elution. Okay, and always remember, suppose you have a test tube having protein and a lot of things, and you want to separate the DNA. What do you add? It is chilled ethanol that you add that separates everything. Works? Ma'am, you're dragon. Okay. Now, heroin. We have come to the next chapter. That is human health and diseases. Heroin is what, my dear children? This would be the last, I think. Let's see. Okay. Heroin is what? Heroin is diacetylmorphine. Okay, heroin is diacetylmorphine. The morphine undergoes acetylation and you get heroin. And heroin, my dear children, is what? It is an opiate. You have three types of drugs. Listen to me carefully. I'm not writing anything now. Okay, for now. That is opiates, cannabinoids and coca-alkaloids. Opiates are the one that affects our CNS, central nervous system, not this, sorry, central nervous system, and also gastrointestinal tract. Two things it affects, okay? And under opiates comes what? Heroin and morphine. Heroin is a depressant. It makes your body depressed, okay? Whereas when you talk about morphine, morphine has two functions. One is it acts as a painkiller, so after surgery you give morphine, and also it is a sedative. Okay, heroin is also known as smack. Heroin is also known as smack. And with that, we complete what? The opiates. And where do you get heroin from? From the latex. From the latex, and also by acetylation of morphine. The product is known as diacetylmorphine, my dear children. Works. The structure of, if in your NCRT, the structure of what do you call opiate is given, and it looks like an O. Always remember, morphine, it is morphine only. It is a chemical structure that is given, and it looks like an O. So you can remember it's from opiate. Now cannabinoids, my dear children, it is going to affect your cardiovascular system. Okay, cannabinoids like marijuana, ganja, hashish and all those things are going to affect your cardiovascular system. Please remember that. And it is the one which is abused by sportsmen. For sportsmen, they use cannabinoids. Okay, they excessively use what? Cannabinoids. That is totally different. Yeah. Now the third thing is coca-alkaloids. Coca-alkaloids means you're talking about cocaine. Where do you get cocaine from? You get cocaine from erythroxylum coca, my dear children. And cocaine is basically going to affect your CNS again, just like opiates. They are also going to affect your CNS, and they are also going to block the receptors of dopamine, which is why you get hallucination, which is why you get euphoria, intense happiness. Apart from them, okay, it is also known as coke and crack also. And apart from this, there are two more things that gives hallucination, that is atropa belladonna and also datura. Two more things, okay? Apart from all these things which are there, you have benzodiazepines and what? Benzodiazepines, and I forgot, barbiturates, amphetamines, three of them. They are there to treat what? Depression and also insomnia. If you're not able to sleep, that is given to you. Okay. Now, one question can be asked: what is that something that increases your blood pressure? It is not cannabinoids. Since it is cannabinoids, you think it is affecting your cardiovascular system, so it will be cannabinoid, but it is not. It is nicotine. Okay. In the tobacco, you get nicotine. Nicotine affects your adrenal glands. Adrenal glands releases adrenaline, which increases your blood pressure and things like that. Okay. Now, one more thing. In the case of sportsmen, one more thing can be given: anabolic steroids. That can also be given. Those are hormones and stuff like that. That is totally different from cannabinoids. Okay, works. Take care. Next thing, human immunodeficiency virus HIV is comprising of what? My dear children, there will be a capsid, protein capsid, and inside the capsid there will be single-stranded RNA. How many of them? 2 of them. So it is not double-stranded, they are both single. Okay, just single, no mingle. So single-stranded RNA, 2 of them are there in the case of human deficiency, immunodeficiency virus. And now one more thing, which is the cell that is the factory? Which is the cell that is the factory of HIV? The factory is macrophage. Okay, which is the cell that is most affected because of HIV? It is T helper lymphocytes. It is what? T helper lymphocytes. Okay, always remember that also. Now, factory and okay, innate immunity, specific type of defense. My dear children, it is non-specific. You have two type of immunity: innate and acquired. Innate is non-specific. Acquired is specific. Okay, innate you have from your childhood. Acquired you get throughout your lifetime. Okay, remember that. So it is non-specific. This is wrong. Malaria could be confirmed by Vidal test. No, Vidal test is for typhoid, and ELISA is for HIV. Remember. Okay, malignant malaria is caused by Plasmodium falciparum. Yes, it is caused by Plasmodium falciparum. Now, active immunity is slow and takes time. Yes, active immunity means you are wanting your body to produce antibodies, so it will be taking time. That is correct. Passive immunity will be fast. Okay, now you might think active is fast. No, passive is actually fast and active is slow. Now, saliva in the mouth acts as a physiological barrier. Yes, my dear children. Okay. Yes, it is going to act like a physiological barrier. So in the case of innate immunity, you have 4 things: physical barrier, physiological barrier, cytokine barrier, cellular barriers. Okay, I think that is the next question also. Yes. So over here, physical barrier, as in you're talking about skin and things like that. Physically, they are going to act like a barrier. Okay, physiological barrier as in secretion. For example, HCl. For example, your lysozyme that is there in your tears. Okay, and also saliva, all of them are physiological barriers. Cellular barriers, as in you're talking about cells. One such cell is natural killer cells, NK cells. After that, you have polymorphonuclear leukocytes. That means you're talking about neutrophils. You're talking about neutrophils. Apart from that, apart from NK, apart from polymorphonucleosides, what do you have, my dear children? You have macrophages. You have monocytes. You have monocytes. All of them are going to be cellular barriers. Cytokine barriers, something that is released by the cells. Okay, those are known as cytokine barriers. Example is interferon. interferon is released by virus infected cell, my dear children. Always remember, virus infected cell release interferons and say that you have to be safe. There is one more interferon given in your NCERT, and that is alpha interferon. Where is it produced? Where it is given, my dear children? For cancer patient it is given. For increasing their immunity, alpha interferon is given. This interferon, that interferon is totally different. Works? Next. Select the correct option. So basically, when plasmodium enters our body, in which stage does it enter? It enters in sporozoite stage, my dear children. The sporozoite stage, when it enters our body, where does it go? Sorry, first of all, in the mosquito, where is the sporozoite present? In the salivary gland. Okay, when mosquito comes and bites, it gives a saliva, and with that, sporozoite comes into our body. Where does it go first? The terrorist attacks first in the liver cells, then it goes to the RBC. In the RBC, what is going to happen? In the RBC, they are going to divide, and they are going to transform into some other stage that is known as gametocyte stage. The gametocyte stage is that stage where they are going to give rise to gametes. But do they form gametes in the human body? No. They're just in the gametocyte stage. So they do that, and also they do one more thing. The plasmodium is going to feed on hemoglobin, and they're going to break down hemoglobin into heme and globin. Heme is kind of very toxic for them, so they convert into what? Hemozoin. They convert into what? Hemozoin. And inside that, what do you have? You have gametocyte stage also. Now what will happen? After some time, this will rupture. Both hemozoin and gametocyte is released. Because of this hemozoin release, this is toxic for us. We get fever and chills in the case of malaria. And these gametocytes, my dear children, are now going to go in the blood. Okay, now are going to go in the blood. Again, mosquito will come and bite. It will take the gametocyte. Inside the gut of the mosquito, the gametocyte will form the gametes. It will fuse. Lots of stages. At the end, back to the salivary gland, sporozoite stage. Works? Now, enzyme which is responsible for replication of HIV in macrophage. My dear children, HIV is what? HIV is having RNA. In our case, in the case of macrophage, a cell will be having what? DNA, right? So, RNA to DNA has to happen. Then only it can come and incorporate over here. So what is that enzyme? This is known as reverse transcriptase. So listen, DNA to RNA is known as transcription. Opposite of it will be reverse transcription. What is the enzyme? Reverse transcriptase. Works? Okay. Now, where will you look for the sporozoites of malarial parasites, my dear children? Okay, this question can come. Where do you look for the sporozoites of malarial parasite? Salivary glands of freshly molted female Anopheles mosquito? Wrong. It is written freshly molted. They have just been formed, so they are not infected. So you have to go for this. Saliva of infected female Anopheles mosquito. There you will be finding it. Okay? Not all mosquitoes have this. Yeah, AIDS is characterized by sharp reduction in, as I told you, T helper cells. Now, yeah, very important. You have two primary lymphoid organs where they are formed. Okay, always remember you have two types of lymphoid organs. One is primary lymphoid organ, other one is secondary lymphoid organ. Okay, now in primary lymphoid organ, my dear children, what is going to happen? They are going to form, they are going to mature, and they are going to become form, they are going to mature, and they are going to become antigen sensitive. They are going to become sensitive to antigen. They are not going to act there. Where do they act? Where do they become effector cells? In the secondary lymphoid organs such as spleen, such as lymph node, such as Peyer's patch, such as tonsils. Okay? Works? So, thymus and bone marrow are basically primary lymphoid organs. In the case of bone marrow is where all the cells are being formed. Okay, B lymphocytes, C lymphocytes, all the cells. But B lymphocytes mature there itself. T lymphocytes goes to the thymus for maturing. So, T lymphocytes mature in the thymus and B lymphocytes mature in the bone marrow itself where they are formed. Okay, now MALT, M-A-L-T, mucosa associated lymphoid tissue. Okay, that is a part of your secondary lymphoid system. Your Peyer's patch, your tonsils, everything is a part of mucosa layer, the innermost layer, right? So, basically, how much percentage do they constitute? 50%. Okay, they constitute how much percentage? My dear children, 50%. Works? Now, next chapter, organism and population. So, children, I have taken first the high weighted chapters of 12th, then we will go to 11th, then we will do the practical questions. Okay, that is our thing. So, over here, match the following. You have mutualism, commensalism, parasitism, competition, predation. Always remember, commensalism is the most easiest to find out. Commensalism is when one organism gets the benefit, another organism is neither harmed nor benefited. Example, epiphytes growing on what? Mango tree. There's a mango tree, epiphyte is growing. Okay, why epiphyte is growing? Because it wants the sun. Is the mango tree affected? No. So, epiphyte gets the profit. So, this will be an answer. Apart from the sea anemone and clownfish example, all these things, cattle egret and cattle egret and what do you call it? Cow. All these examples are there. Parasitism is basically when one is getting affected and other one is getting profit. Okay, ticks and the dogs. On the dogs you might find ticks, and this is example of what? Parasitism. Competition is basically when both of them are fighting. Yeah. So competition is found in belanus and thalamus. Fine. And when, listen to me carefully. In competition, if both of them are fighting, what is going to happen? Competitive exclusion is going to happen. They are going to be moving out. Second question, if they want to coexist, what is the thing that they can have? Resource partitioning. If they want to coexist, they have to do resource partition of the resources. For example, you and me want a cake. Why not divide into two and give to you and one to me? Resource partitioning. Okay, that is for coexistence. And now, suppose because one superior variety is removed, what will happen? What do you call this process when the other organism will be moving to the broader area? This is known as competitive release. Three things. Okay. Now, mutualism, my dear children, where both of them are going to be benefited. It is orchid, ophrys and the bees. So, orchid, ophrys and the bees, people confuse it for parasitism, but it is not. Always remember, the bee thinks that the flower is like a female bee, so it tries to copulate. It is known as pseudo-copulation. But the orchid just wants pollination to happen. So, basically, both of them are co-evolving. They show mutualism only. Okay, they show mutualism. Predation is sparrow and the seed. Next thing. In accordance with the lifestyle, parasite evolved special adaptation. Which of the adaptations are correct? High reproductive capacity, yes, they want to increase the amount. So yes, that is there. Highly advanced digestive system, my dear children, no, they have reduced digestive system or they have no digestive system. Okay, so this is wrong. Loss of unnecessary sense organs, obviously, suppose they have ears, can they move around? No, so that is also correct. Presence of suckers, that is also correct. Okay, only this one is the wrong one. Next thing, okay. I told you, if at any point species facing competition might evolve mechanisms that promote coexistence. This is known as resource partitioning. And now, over here, very important, very important. Age pyramid, my dear children, which one do you think will show increasing human population? So, the lower region is the pre-reproductive organism in the pre-reproductive area. That means they will be reproduced. They are small now, they will grow and reproduce later on. Okay, then comes reproductive area and then comes what? Post-reproductive, that means the old people. So, if you see the broadest version is the lowest version, that means human beings are going to be, the human population is going to be increased. So, over here it's almost same, so I cannot take this, but over here I see it's increasing. So, this is the one that shows that human population is increasing. Now, plus sign if it is assigned to beneficial interaction, minus is for negative and O is for neutral. Then what do you mean by plus and minus? Where do you see it? In mutualism it is plus and plus. Okay, in amensalism it is what? Minus. It is minus and 0. One is going to be badly affected and otherwise is going to be neither harm nor affected. Okay, commensalism is positive and minus, positive and 0, and parasitism is this. So the answer becomes this. Okay. Next question. Okay, very important question. Select the correct option. So with this, you get answer to a lot of questions coming up from this graph. Listen to me. So this is population density, this is time. Okay, you have a graph like this. What does this show? Exponential growth. It is just growing in a constant rate, my dear children. It is assuming the factors are not limiting. There is enough amount of food, there is enough amount of water, everything. Okay, this is known as exponential growth. Okay, and this one, where basically there is a lag phase and it slowly goes off and you reach over here. Until here, you do not go up. Why, my dear children? Because the resources are limiting. This is known as logistic growth, logical growth, yeah, and logistic growth. And basically, there's an S-shaped curve. This is a J-shaped curve. This is an S-shaped curve. And what do you call this condition? Over here, when it reaches, it is known as asymptote. Okay, what do you call this? Asymptote. Even if you do not remember, it's okay. Remember S-shaped for sigmoid, yeah, and this one exponential. And remember the formula. So, dn by dt. My dear children, for this one, okay, for exponential, it is dn by dt is equals to Rn. What do you mean by R, my dear children? Intrinsic rate of natural increase. R suggests B minus D. That is birth and death rate. Okay, that is what it suggests. Now going ahead, my dear children, you have small b. What is this for? What is this for? This suggests what? dn by dt is equals to Rn k minus n divided by k. What does k suggest? K means carrying capacity beyond which the resources can be, the organism cannot be growing. So please remember, for this is this. Okay, for this you have this. They can ask you questions based on this. Now, statement, predators help in maintaining species diversity in the community. Yes, predators help. Suppose the prey population is going on increasing. Would they allow other population to grow? No. So yes, species diversity increase. You need predators. Second thing, it reduces the intensity of competition among competing prey species. That is also correct. Work. Now, okay. Evolution chapter, okay, evolution chapter. Children, I'm going to pick certain questions, I'm going to explain and then we'll move ahead. Don't worry. So, what is the chronological sequence of human evolution? First thing, you had Dryopithecus and Ramapithecus. Dryopithecus, Ramapithecus. Dryopithecus were more aim-like, Ramapithecus, Ram. They were more man-like. From Ramapithecus came Australopithecus, okay. From Australopithecus came what? Homo habilis, Homo habibi. The first human-like features were found in there and they had 650 to 800 cranial, 650 to 800 cranial capacity, that is correct. And also they didn't eat meat, they ate fruits. Okay, they ate fruits, always remember. So after Ramapithecus, Australopithecus, and then you have Homo habilis. Until then, they only ate fruit, nothing else. So they were excellent tool makers, but never touched meat. From Homo erectus, they started touching meat, my dear children. From Homo erectus, your brain capacity increased to 900 cc. Then you came to Neanderthal man. Neanderthal had a brain capacity of 1400 cc, 1400 cc, my dear children. And also, they were the first one to protect their body, cover their body, and also bury their dead. Okay, they started burying their dead. And at last, you got Homo sapiens, which is us. Okay. Now, what do you mean by saltation? Saltation is single step large mutation. That is what you mean by mutation. Saltation. Now, listen to me. There are two things, Charles Darwin and Hugo de Vries. Charles Darwin said that basically variations are going to be directional. That means it is going to be always in favor. Always remember, variations directional, given by Charles Darwin. But Hugo de Vries says variations arises because of mutation and they are directionless. Any type of mutation can happen, good or bad. Directionless was given by Hugo de Vries. Do not forget. And which of the following is low-finned fish? So you had fishes. From fishes came amphibians. What is the connecting link that you have? It is known as low fins or known as coelacanth. So they had fins, but with that fins they could walk around. So they are known as low-finned, or else it is known as coelacanth also. That is the connecting link between Pisces and amphibians. Going ahead, the idea of mutation was given by Hugo de Vries, who worked on what? Evening primrose. He was working on evening primrose. He said mutations are what? Directionless, my dear children. Going ahead, all the facts are true about Neanderthal man, except I told you 1400 cc is correct. Use height and lived. Yeah, this is also important. Lived 1 lakh to 40,000 years back. Okay, and their fossils were discovered in Java in 1891. Wrong. The Java, this is found in Homo erectus. This is about Homo erectus. Homo erectus are also known as Java men. Okay, works? Okay, next chapter, biodiversity and conservation. Given below is the representation of extent of global diversity of vertebrates. What groups does the portion include? This is the last chapter from 12th I am going to discuss. Now we are going to move. Okay, so over here always remember, in the case of vertebrates, the maximum amount is fishes. Is what? Fishes. After fishes come mammals, FM. Radio FM, you know, right? FM. You remember like that. Then come birds. Then you have reptiles and amphibians. Birds also have enough amount of portion. Birds also have enough amount of portion. So fishes and then birds. FB, Facebook, both of them have enough amount of portion. And also it is mammal in between, and here reptiles and here amphibians. This is the thing. In the case of invertebrates, what are the most most highest thing? You have insects. Insects are found in the most highest amount. Now, very important question asked so many times over here. Dodo is from what, my dear children? Dodo is from Mauritius. All of them are extinct species by IUCN. Dodo is from Mauritius. Quagga is from what? Easy, I'll tell you. Dodo is from Mauritius. Steller sea cow is from Russia. Okay, then you have thylacine and then you have quagga. So basically over here, okay, thylacine goes with Australia and quagga goes with Africa. Is that correct? Is that correct? Yes, yes. Okay, so always remember this thing, and also remember one more very important thing, that is basically there are two organisms which got extinct because of over exploitation, and those are Steller sea cow and passenger pigeon, not them. Okay, Steller sea cow and passenger pigeon. Remember, this is very, very important. Dodo from Mauritius, quagga from Africa, thylacine from Australia, and Steller sea cow from Russia. Okay. Next thing, India is one of the 12 megadynasty countries with dash of the genetic resources of the world, 8.1%. Okay, 8.1% of the genetic resources of the world. Remember that. Tropical rainforests were occupying almost 14%, but now it's just 6. So 14's half is 7. So you can remember 6 is nearest to that. 14 and 6. Okay, and here it is 8.1%. Going ahead, what do we have? Initially A biodiversity hotspots were identified, but subsequently B have been added to the list. So initially 25 were there, and then you added 9, and then you got 34. So initially 25, if you remember that is also fine. 25, then plus 9, you got 34. Biodiversity hotspots. Now, my dear children, when you are exploring molecular genetic species level diversity for products of economic importance, what do you call this? You call this as bioprospecting. Okay, what do you call this? Bioprospecting. Now, there is an equation for species area relationship. What is the equation? At least remember that. It is S is equals to C A to the power Z. Okay, species richness is proportional to area, but up to a limit. That means, for example, if I explore more area, I will be able to see more species. But can it go on like that? No, my dear children, up to a certain limit, which is why it is represented by this. Now, log equation for the same is log S is equals to log C plus log A. Where do you put the Z in front? Okay, this is the logarithmic equation. What does C stand for? C stands for the y-intercept. Okay, and what does Z stands for? Z stands for the slope of the line. This is the slope of the line, and where it touches, it is known as the y-intercept. Works? Now, sacred groves, my dear children, again very important thing. Sacred groves are areas which I am going to protect because I have some religious attachment to that. Okay, so it comes under in situ conservation only. Always remember, wildlife sanctuaries, national parks, biodiversity reserves and sacred groves are going to come over here. Wildlife, wildlife safari is different. That is the only thing that is ex situ conservation. This wildlife sanctuaries comes under in situ only, just like sacred groves. So Kashi and Jaintia of Meghalaya, Aravalli Hills of Rajasthan, Western Ghats of Karnataka and Maharashtra, and also Chanda, Bastar and Sarguja area of Madhya Pradesh. Also remember one very important thing, that is in Meghalaya, this is the only source of safeguarding lot of things. Okay, works. Now, my dear children, penicillin, it is produced by what? Penicillium notatum. Citric acid produced by Aspergillus niger. Butyric acid by Clostridium botulicum. Acetic acid by Acetobacter aceti. And lactic acid by Lactobacillus. Okay. Children, your exam starts from 2. What is the correct timing? Can anyone tell me? 2:30 is what I know. Yes. Okay, 2:20 you have to enter the hall, right? Yes, 2:20. Okay. So we'll do fast. Okay, chalo, listen to me. Over here, one more thing, that is citric acid is produced by Aspergillus niger. This is the fungus. Even Penicillium notatum is a fungus, and penicillin is what? Antibiotic. Now over here, what are flocks, my dear children? Flocks are basically the mesh-like structure which is formed with the help of aerobic bacteria and also fungal filaments together. Okay. 2:30, but we have to be there early. Yes, sure, my dear children. So those of you who are just there going for bio examination, you can go ahead. Okay, works. And over here, listen, statin. Statin is for stabilizing the blood cholesterol level. Cyclosporin A is for what? Cyclosporin A is for what do you call this? It is an immunosuppressive agent. Yeah, you get it from Trichoderma polysporum. Streptokinase is a clot removal. Okay, it is for clot buster. And lipase is basically for removal of oil stains. Pedigree chart. From here starts the practical questions. Listen to me carefully. Pedigree chart, how do you analyze it? If, my dear children, at the top, one parent is affected, okay, at least one parent is affected, that it has to be dominant. It has to be a dominant disorder. Over here, no parents is affected. In the parents, it is not found, but in the progeny, it is found. It is a recessive disorder. It will be a recessive disorder. How do you identify whether it's autosomal or sex-linked? If both the progenies are equally affected, male and the female, it is known as autosomal. If both the male and the female, okay, are equally affected, it is known as autosomal disorder. So both of them are going to be autosomal. Okay, this is going to be autosomal dominant and this is going to be autosomal recessive. This one is myotonic dystrophy given in your NCERT, okay, and this one is sickle cell anemia, thalassemia, or it can be anything else. Okay, even phenylketonuria also. Now, in the case of sex-linked, my dear children, you will find that more males are affected. That means the boxes would be colored. Male boxes would be colored. Works? Now, which of the following is an example of multilocular ovary, my dear children? Multiple locules. Where do you find it? You find it in tomato. This is the example. So when the placenta is axial, okay, this is axial placentation, my dear children, where you have this like this and stripes like this. Okay, this is axial placentation. Here you will be finding multilocular ovary. This is found in tomato. Next, which one of placentation is found in pea and bean? So over here, marginal. At the margin, you find the placentation. This is known as marginal. So pea and bean have marginal placentation. Remember that. Second thing, in which of the following plants is axial placentation observed? Again, you're talking about axial placentation. One, you talk about tomato. Apart from that, you have in hibiscus also. You have in hibiscus or China rose. So China rose, hibiscus and lemon, all of them are example of axial placentation. In between you have it. These are the ovules and they have lines. Okay. Next, parietal placentation. Parietal means towards the periphery you will find placentation. Where do you find it? In mustard. In mustard. So MP you can remember like that. Okay. Now, going ahead, my dear children, free central placentation is characteristics of. Free central means in the center you find it. Okay, where do you find it? In dianthus. In dianthus, my dear children, you will be finding free central placentation. Cool. Next thing, which of the following exhibit basal placentation? At the base side, you find placentation. Marigold and sunflower. Marigold and sunflower. We'll go back again once more. Listen to me carefully. Basal placentation in marigold and sunflower. Okay, and then free central placentation in dianthus. Parietal placentation outside, you have mustard. Okay, and axial placentation in hibiscus, and marginal placentation in what? Pea. And axial presentation, you have China rose and lemon also as an example. Going ahead, what are the two cells present in the pollen grains, my dear children? Generative cell and vegetative cell. Going ahead, which specimens are commonly used in the lab for studying pollen germination? It's Windsor roseae. It is what? Windsor roseae, because it has high viability of the pollen grains. Going ahead, which solution is used to nurture pollen grains, my dear children? It is sucrose solution. Why not anything else? Because sucrose is going to provide the same osmotic balance. The pollen grains are not going to lose water and things like that. It is like the same osmotic balance, and you need energy. If you put glucose, it won't be giving you normal osmotic balance. So it is sucrose, 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 sucrose. Okay, which chemical is found in the exine of the grain? It is sporopollenin. In nature, where is the nutrient medium for pollen germination present? You are providing it now, right? But in nature, where is it present? On the stigma. There, that's where it comes and lands, right? Now next thing, what does the pollen tube carry? It will carry two male gametes, and I told you one tube nucleus. Tube nucleus belongs to the vegetative cell. Going ahead, what is the correct formula to calculate percentage of pollen grain? So N is the number of germinated pollens by total number of pollen grains that is observed into 100. N, small n, by capital N into 100. That will be the formula. Now, which stain is used to observe pollen tube growth, my dear children? It is cotton blue. If you want to observe pollen tube growth, you use cotton blue. Cotton blue stains the pollen tube wall, making it directly, because it can bind to what? Cellulose and pectin. That is why. Why not saffron? Because it is going to stain lignified tissues. Okay, that is not found in pollen tube. Okay, methylene blue is for staining the nucleic acids. Okay, that is your DNA and things like that. Acetocarmine is there for staining what? The chromatin fibers which are there. So for mitosis, what do you use? You use acetocarmine. Okay, for staining the pollen tube growth, you use cotton blue. Now, can pollen grains of one plant species germinate on the stigma of another? No, because pollen pistil interaction is what? Species specific. Going ahead, which of the following is not a substage of interphase? In interphase, my dear children, you have three stages: G1, S phase, G0. G1, S phase, G2. Okay, G1, S phase, G2, right? In G phase, the cell grows in size. Some organelles, they replicate. In S phase, it is known as synthesis phase, because the DNA is going to replicate, and also the centrioles are going to replicate. And then you have G0 phase. What happens in G0 phase, my dear children? In G0 phase, the other organelles which are there, they are going to replicate whichever is left behind, and also the proteins also replicate. They also increase in number. But telophase is not going to come under that. Telophase is what comes under mitosis. So listen to me carefully, okay? You know, G1, S, wrong. You have G1, you have S and G0. Okay. Now over here you have M phase, where the actual division takes place. In M phase also you have two phases. Karyokinesis, where the nucleus divides, and cytokinesis, where the cell is going to divide. Okay, in karyokinesis you have prophase, metaphase, anaphase, telophase, and then in cytokinesis the cell is going to finally divide. And there is a G0 phase. Okay, the cells which are entering in the G0 phase. G0 phase means they do not want to undergo division. They're just there. In G0 phase, you say cells are metabolically active. Remember that. They are what? Metabolically active, my dear children. Remember that. Okay. Now, what is the preparatory phase of mitosis? Preparatory phase is interphase, my dear children. This is the interphase. This is where they prepare themselves. And this has 23 hours. If for 24 hours is the time for cell division, 23 hours they spend over here. 1 hour they spend for division. Okay. Now, if you are asked about the synthesis phase, it is S phase. Now, what major event takes place in the S phase of interphase? Replication of the DNA and also of the centrioles. Okay. Ma'am, order of the interphase, I told you, G1, S, G2, G1, S, G2, and over here if you're going out, G0, it is metabolically active there. Okay, such as your heart muscle cells, okay, they are metabolically active, they are in G0 phase. Now, I told you this. Which stain is commonly used to observe the stage of mitosis? Acetocarmine. Okay, now what are the two major steps of mitosis? Karyokinesis and cytokinesis. Karyo means nucleus division, cyto means cell division. Going ahead, primary producers of pond ecosystem, my dear children. It can be phytoplankton, submerged aquatic plants, floating aquatic plants, rooted angiosperms, all of them can be primary producers. Going ahead, rate of formation of organic matter by rabbit in grassland is known as. Rabbit is what? Not the producer, but the consumer. So when the plants produce something, you call it as primary productivity. Now when the organism feeding on it produces something, you call it secondary productivity. Okay, always remember that. Now, yeah, very important thing. Which of the following representation shows a pyramid of numbers in forest ecosystem? In forest, you have so many trees, my dear children, and on that you have insects, you have other organisms, but the top level organisms, they would be less. So I'll say this is the graph. This is the graph. If you're talking about just one tree, then it would have been opposite. One tree means number of trees just one. Yeah, insects would be more, and this would be the graph. But you're talking about the whole forest, then it will be this. Works? Now, annual net primary productivity of the whole biosphere is 170 billion tons. It's billion. Okay, B. B comes before M, right? So always remember, it's 170 billion tons. That is the net primary productivity. Decomposition rate is going to be slower if what? If the things that we are going to decompose, it is rich in lignin and chitin, because it is difficult to break them down. So what is the perfect atmosphere for decomposition to happen? It should be warm, it should be moist condition, it should be promoting, warm and moist condition promotes decomposition. There should be air, because the aerobic bacteria is going to break things down. So there should be air, yeah, that means oxygen should be there, and there should be high temperature, not low temperature. So warm, moist, high temperature and aerobic condition. Okay, warm, moist, high temperature, aerobic condition, not anaerobic condition. Now, the standing crop, what do you mean by that? Standing crop is basically what? The measured as the mass of living organisms or the number in a unit area. So mass of living organism, biomass, the number in unit area, all of them is correct. That is standing crop. Going ahead, find out the correct sequence of stages involved in decomposition. Very important. First thing, you have to break down big things into small things. So fragmentation. Okay, earthworms, detritivores are going to eat the detritus. They're going to poop things out, break big things into small things. Yeah, and then from the small things, the water soluble inorganic things are going to leach to the soil. And then what will happen? Now we want to break down organic matter into inorganic. This is known as catabolism. Organic to inorganic. And when you do that, it's not a one-step process. When you break down organic to inorganic, in the middle you get humus. This is known as humification. This process until halfway it is known as humification. Now, from this humus, when the inorganic nutrients are going to release slowly, this is known as mineralization. So first fragmentation, then leaching, then catabolism, humification, and then mineralization. Works? Now, select the, okay, now starts the 11th. Okay, are we ready for the 11th? Yeah? Select the correctly matched pair organism with their order. Mangifera indica, primata, wrong. Primates come under mammalia. Triticum aestivum, sapindales, wrong. It is povales. Musca domestica, my dear children, it is diptera, correct. Your makhi, what do you call fly. Homo sapiens, my dear children, they come under primates. Next thing, in taxonomic hierarchy, the number of common characters will increase when we go from, always remember, king, Peter, came, over, for, a great, soup. Yes, so basically when we go down, the common character is going to increase. In species of humans, my dear children, we are all similar. We all have hands, we all have lengths, right? So this is where it goes on increasing. So kingdom to species. Cool. Next, match the following classes of fungi with the examples. So over here you have the examples. Phycomycetes, my dear children, will go with albugo. Albugo, always remember, it is the one that causes white rust in crucifers like mustard. You might be asked this question. Albugo is what, my dear children? It is what? A parasite. Okay, it is a parasite. Ascomycetes, you have penicillium that produces penicillin. Okay, basidiomycetes, what does it? It is Puccinia, my dear children. Puccinia is again going to cause problems in the case of wheat. Rust disease in what? Wheat. So white rust, this is albugo. Puccinia is going to cause rust in what? Wheat. Now deuteromycetes, you have alternaria. That is a separate thing. It is known as imperfect fungi because it does not have, deuteromycetes does not have sexual stages. They have asexual stages. Remember that. Okay, works. Next thing. Which of the following protozoan parasite cause sleeping sickness? Plasmodium, Entamoeba, Leishmania. Plasmodium causes malaria. Entamoeba causes amoebiasis. Leishmania causes leishmaniasis. Trypanosoma is the one that causes sleeping sickness. Why? Trypanosoma, my dear children, going to affect the neural control system. That is why you have confusion and also sleeping disorders. Okay, and it is transmitted by sassafly, Glossina species. Okay, next thing. Match the column 1 with column 2 in correct options. Bryophyta, gymnosperm, algae and pteridophyta. What comes under what? Bryophyta, my dear children, you have sphagnum. Bryophyta is what? Sphagnum, and sphagnum is what? Unicell. No, leave that. Gymnosperm, my dear children, gymnosperm are basically the one that bears the naked seeds. And what do you have over here? You have pinus over here. Then you have algae. Algae is ectocarpus, yeah. And pteridophyta, you have ediantum. Yes, is there any feature that I want to tell you? Let me see. So bryophytes, my dear children, they are non-vascular obviously, and they need water for what? Reproduction to happen. Okay, they need water for reproduction to happen. Gymnosperms, my dear children, they have naked seeds, and as I already told you, algae, ectocarpus, this is brown algae found in the marine environment, and pteridophytes, my dear children, the first one to have what? Vascular system. That is xylem and phloem and everything out there. Okay, cool. Let's go ahead. Come on this side. Yes. Different types of respiratory organs like gills, book lungs and trachea are found in what? Arthropods. All arthropods are the species, most diverse organism, right? So obviously they would be having what? They would be having different type of lungs also. You can think like that. Okay, so it is arthropods. Now going ahead, okay, one more thing. Where do you find radula, rasping organ? That is found in mollusca. That is found in what? Mollusca. Now observe the following and tell the scheme. So vertebrata, my dear children, you can divide into two things: agnatha and gnathostomata. Agnatha means they do not have a jaw, so they have a mouth with teeth just like arranged like that. They do not have a jaw. And then you have what? Gnathostomata, where you have a mouth with jaw. Okay, gnathostomata. Under that you have something that bears fins, something that bears limbs. Fins are found in what? Fins are found in fishes and limbs are found in tetrapods. We have four podiums, right? So these are what? Tetrapods. So first is agnatha, this is gnathostomata. Under gnathostomata you have what? Pisces and tetrapoda. Fine. Now, which of the following phyla possesses body cavity as shown in the diagram? Very important thing. We have different type of body cavity, my dear children. If it is shown like this, so this whole thing is covered. That means it is acoelomates. They are what? Acoelomates. Example, porifera. Okay. Example, porifera, then nidaria, then ctenophora and platyhelminthes. Until platyhelminthes, my dear children, all of them are acoelomates, do not have coelom. After that comes aschelminthes. They definitely have a pseudome, but it is pseudocoelom because it is scattered like this and is not entirely covered by mesoderm. Only if the cavity is covered by mesoderm, you call this as what? Proper coelom. But they are pseudocoelomates. What are they? Pseudocoelomates. And then you have one more thing. Okay, you have one more thing that is known as coelomates. So annelida to chordata, all of them are going to be coelomates. So please remember, until platyhelminthes, acoelomates, then comes pseudocoelomates, that is aschelminthes, and then comes coelomates. Now, okay, choose, match the animals of column 1 with the respective classes in column 2. Aptenodytes. Aptenodytes is what? Penguin. Yes, it is what? Penguin, my dear children. And penguins comes under, penguin is a bird? Penguin is a bird, yes. It comes under apes, right? Now, Hemidactylus, basically talking about lizard. Lizard comes under reptilia. Carcharodon, my dear children, it is what? It is the great white shark. And sharks come under what? Pisces. And in Pisces also, sharks are under chondrichthyes, cartilaginous fishes. So Carcharodon is basically shark and it comes under chondrichthyes. Pteropus, the flying fox, or you call it as a bat, and bat comes under mammalia. Bat comes under mammalia. Got it? Cool. Next, identify the phylum X. Okay, phylum X. You have Animalia. Okay, they are under Animalia. They have tissue grade of organization. My dear children, only two organisms have tissue grade organization. One is Cnidaria, another one is Ctenophora. Okay, none of the others. Now, bilateral, that is wrong. It should be radial. Okay, they are radially symmetrical. That means they can be cut into equal halves from any plane passing through the center. That is radial symmetry, and that is found in both jellyfishes, that is Cnidarians, and also the comb jellies, that is Ctenophorans. Okay, and they are acoelomates as well. So over here you just have Ctenophora, so you go with that. And I'll tell you one more thing. So asymmetrical are poriferans. Okay, radially symmetrical are them, that is Cnidarians, Ctenophorans, and also one more thing, the adult form of echinodermata, for example starfishes. Okay, but my dear children, the larval form of echinoderms, they are bilaterally symmetrical. So adults are immature, they are radially symmetrical, but the younger ones, the larval forms, they are bilaterally symmetrical. Bilateral symmetrical is something that you found in big organisms. So from them, from platyhelminthes onwards, my dear children, all of them are bilaterally symmetrical. Okay, all of them are going to be bilaterally symmetrical except echinoderms, whose adult form is radially symmetrical, but larval form is, again, as I said, the symmetrical. Now, identify the floral formula of plant belonging to potato family, my dear children. So in potato family, potato family is basically Solanaceae family. Solanaceae family. Always remember Solanaceae. That is where potato belongs to. So they are having bisexual flowers. Yeah, calyx is there. Okay, they have the sepals, and all the sepals are fused together like this. That is why they are inside the bracket. Then you have corolla. That is the petals. Again, 5 of them fused together, which is why they are in the bracket. Then you have androecium. How many? 5 of them. The male's reproductive part, 5 of them are there. And basically, I tell you, if suppose this is a petal, basically from here only the filament is arising. That is, the male part is arising. This condition is known as epipetalis condition. Both of them are linked together. That is why you see this. Okay. Then gynoecium. If you talk about gynoecium, it is hypogynous. What do you mean by hypogynous? The ovaries at the top. Every part else, the petals and everything, they are at the bottom. So this is known as gynoecium. This is known as superior ovary, which is why it is above the dash. And how many of them are there? 2. So 5, and 2. Okay, androecium is not fused together because they are on the petals, right? So they cannot be fused. Gynoecium is fused. How many? 2 of them. Works? You are more faster than this, I cannot do. Epigynous flower is one in which epigynous, my dear children, where the ovary is inferior and the other floral parts are superior. That is what you mean by epigynous flowers. Okay, what is perigynous? Just a second, I have a confusion with that. Okay, perigynous is basically Okay, this question I'll leave for some time. I'll come back to it. Okay, before the class ends, I'll tell you. In plants, lateral roots arise from. Lateral roots, my dear children, where do they come from? You have epidermis. You have, after epidermis, you have the cortex and a lot of other things. Yeah, endodermis, and after that comes pericycle, and from pericycle you get what? Lateral roots. From pericycle you are going to get lateral roots. Thank you so much for children who are saying thank you. Yeah. Now identify the part labeled as M. So if this is a chromosome, at the top you see these portions. These are known as satellite. This is known as satellite. What do you call this? This is known as centromere. What do you call this? This is a chromatid. This is a chromatid. This is a sister chromatid. Okay. Going ahead. Oh my God, it went again there. Now, identify the major site of biosynthesis of lipids. My dear children, always remember, RER, rough endoplasmic reticulum, has ribosomes, and ribosomes are there for the production of proteins. Yeah. So in RER you get proteins. What about SCR? SCR you only get lipids. So proteins and lipids are formed. Now they go to what? Golgi apparatus. In Golgi apparatus, modification happens. They become glycolipids and glycoproteins. So if you're asked about glycolipids and glycoproteins, it is Golgi apparatus. If you're asked about lipids, it is SCR. If you're asked about protein, it will be RER. So here it's asked about lipids, so it will be SER. Works. Next thing. Match list 1 and list 2 with respect to proteins and the function and select the correct options. Collagen, trypsin, insulin, antibody. Trypsin is an enzyme that breaks down proteins. Okay. Insulin is a hormone that basically converts glucose to what? Glycogen. Glucose to glycogen. So it helps in the process of glycogenesis. Always remember, insulin will be favoring glycogenesis, glycogen formation. Okay. If it is written in the option that it inhibits glycogenesis, it will be wrong. Okay, now collagen is basically intercellular ground substance. In between the cell, you have collagen. Antibody is what it fights against the infectious agents. Going ahead with the next question that we have, match the compounds. Trypsin, as I told you, it's basically enzyme. GLUT4 enables the glucose transport into the cell and all the things. Same. What is the role of competitive inhibitor during enzyme action? Always remember, one famous competitive inhibitor, malonate. Malonate basically is succinic dehydrogenase. Okay, there is an enzyme that is succinic dehydrogenase, and malonate acts as a competitive inhibitor in that case. What happens? Basically, this particular malonate comes and tries to attach to the active site instead of the substrate, and does not allow the substrate to attach to it. So this is known as competitive inhibition. And what do? It declines the enzyme action. Obviously, it declines what? The enzyme action out there. Fine. Next thing. The complex formed by a pair of synapse homologous chromosome is known as bivalent. Can you listen to me? Can you spare at least 1 minute? I will tell you the whole prophase stage. Okay, listen to me carefully. Lazy zebra play during day. Okay, in the case of prophase 1, my dear children, what happens is that in leptoid, the chromatin fibers which are there, they are going to condense down to form the chromosomes. Okay, they are going to form the chromosomes. Now what happens in zygote stage? Basically, suppose I am getting 1 chromosome from my father, 1 chromosome from my mother. This is how it usually happens, right? Now before prophase, what was there? There was G2 and M phase, so G2 and S phase. In S phase, the DNA got duplicated, which is why it looks like this now. It looks like this. Now, how can I make the 2 chromosomes come together? When do I call them homologous chromosome? That is the first question. I call them homologous chromosome basically when from the father's chromosome on the same position I have a gene for height. Here also I have a gene for height. So these chromosomes are known as homologous chromosome, my dear children. So in the case of zygote, okay, both the chromosomes are going, homologous chromosome are going to come together, and here is a Feviquick-like structure being formed that is known as synaptonemal complex. Synapsis is going to happen over here. Yes. So synapsis is going to form over here. Okay, synaptonemal complex is going to form over here. After that, in the case of picotin, what will happen? Crossing over. They are going to cross over. They are going to exchange their material. This is known as crossing over. And which enzyme plays a major role? Recombinase. So in picotin, my dear children, in picotin, you will be finding crossing over. And also with the enzyme what? Recombinase. Now in diplotin stage, this is diplotin stage, okay? They want to break up. When they try to break up, my dear children, they are actually attached to each other at one particular point. There is an X-shaped structure being formed. What do you call this? Chiasmata. They found that they are attached with the help of what? Chiasmata. Okay, and lastly in dikinesis, they are going to finally move away. Can they move away? No. Basically the chiasmata moves to terminal position, so I will say terminalization of chiasmata takes place over here. Now this condition where you have 2 chromosomes like this, you call this as bivalent condition, or you also call it a tetrad. 1, 2, 3, 4. This is known as tetrad. Okay, and always remember, recombination happens between non-sister chromatids. This is non-sister chromatid. They both are sister chromatids. Works. Okay. No. Okay, I also told you synapses, crossing over, chiasmata and terminalization. Now, cells in the quiescent stage, my dear children, they remain metabolically active. Now, what is the function of enzyme recombinase? Helps in crossing over between non-sister chromatids, not sister. Now, in the maize plant, CO2 fixation occurs in both mesophyll and bundle sheath cell because that is a C4 plant. Okay, this is a C4 plant. In C4 plant, my dear children, CO2 fixes twice. This in the mesophyll, this is in the bundle sheath. Here, in the case of C4 plants, photorespiration occurs more or less. It will be less. C3 plants more. Why? Because there is no segregation like this. What do you call the segregation? Kranz anatomy. Over here, PEP carboxylase fixes the carbon dioxide first, and then over here Rubisco fixes it again. Rubisco will be fixing it again in the case of maize plants. Okay. Now the outcome of Calvin cycle includes 1 glucose, 18 ATP, ADP not ATP. Okay, you are utilizing the ATP, and 12 NADP. Again, you are utilizing NADPH also. So 18 ADP, 12 NADPH, and 1 glucose. If you are talking about 1 carbon dioxide, 3 ADP, okay, and 2 NADPH is being utilized. Okay, the outcome is what? You utilized it and now you are getting this. This is what you get. Now, next thing, statement A, photorespiration decreases photosynthetic output. Obviously, yes, yeah. In photorespiratory pathway, neither ATP nor NADPH is produced. Yes, it is not. Instead, it is being utilized. This is correct, this is correct. Always remember, photorespiratory is energetically effective. They take in all the ATPs. We do not want to do that. Okay, it is not going to produce both of them, it is going to take in that. Next thing, respiratory coefficient of glucose, my dear children, it is 1. It will be 1. How do you calculate it? Volume of CO2 evolved divided by volume of oxygen consumed. Okay, volume of CO2 evolved divided by volume of oxygen consumed. By 6 by 6, it will becomes 1. So 1 is the respiratory coefficient of glucose. You can be asked about fat or lecithin and things like that. It is 0.7. It is what? 0.7, not 1. Now match the location of the cell. Mitochondrial matrix, my dear children. What happens in the mitochondrial matrix? What happens? Krebs cycle happens. Yeah. So basically, first thing, in the cytoplasm, glycolysis happens, glucose to pyruvate conversion happens, pyruvate gets into the mitochondrial matrix. In the mitochondrial matrix, basically this converts into acetyl-CoA, and the acetyl-CoA will do the Krebs cycle, form GTP, NADPH, and all this thing, FADH2 and all this thing. And now basically with the help of ETC cycle, they are going to transfer the electrons, which are, so these enzymes are present in the inner mitochondrial membrane. The ETC cycle happens in the inner mitochondrial membrane, my dear children. And in the mitochondrial membrane, you have F0 and F1 particles which help in ATP synthesis. F0 in the membrane, F1 in the matrix. Okay, it is known as, children, it's going to get over. Fast. Okay, over here, glucose to pyruvate. Pyruvate will convert into what? First acetaldehyde, then to what? Then to ethanol in the case of yeast. Okay, so not directly ethanol, first acetaldehyde, then ethanol at the end. And over here it produces CO2 as well. That is a question. Next, match the compounds. So first of all, oxaloacetate is a 4-carbon compound, my dear children. Acetyl coenzyme A, it is 2 carbon, 4 plus 2, it gives you citric acid, which is 6-carbon compound. Always remember that. Ribulose bisphosphate is a 5-carbon compound. Phosphoenol pyruvate is what? Phosphoenol pyruvate is a 3-carbon compound. Okay. Now, carnitine is a derivative of adenine, which is pyrimidine. Wrong. Carnitine is a derivative of adenine, but it is purine. Now, technique for decapitation, that means you're cutting off the topmost portion. It is used in tea plantation. Yes, my dear children, because you want lateral growth to happen. Ethylene is a gaseous hormone. Correct. Use of GA3, that is gibberellic acid. The Hastings, that means increases the malting process in the brewing industry. That is also correct. ABA is a growth promoter. Wrong. ABA is a growth inhibitor. Okay. Next thing, in the standard ECG, I'll tell you this. Now, the number of cells, if you talk about neutrophils, they are going to be the highest. Always remember that. It is going to be 60 to 65. Basophils are going to be the lowest. Base, okay, base. You can remember like that. It can be 0.5 to 1, okay. Just after basophils, you have eosinophils with 2 to 3. And then lymphocytes, if you think, they are comparatively more, they're 20 to 25%. Fine, and monocytes will be 6 to 8%. So neutrophils at the highest, basophils at the lowest. Okay, lymphocytes basically 20-25. Remember that. Now choose the correct statement. Erythroblastus fetalis may result when the fetus is Rh negative and the mother is Rh positive. Wrong. The mother will be Rh negative and the baby would be Rh positive. Okay, remember. Works? Cool. So I think last thing for those who are there, okay, this is atrial, atrium contraction, okay, both the atrium contracts over here. QRS is when ventricles contract. Okay, and T is when ventricles relax. So where does atrium relax? Atrium relaxes over here. So you call this T as repolarization of ventricles. You call contraction as depolarization. So here depolarization of atrium, here depolarization of ventricles, here repolarization of ventricles. Works. Got it? You are most welcome. I think, children, this is the last question. Should I do it? Just listen to this question also. Okay, contraction happens when two Z lines come together. Okay, and in between you have the M line. So Z and M comes closer. Where is the sacromere? In between two Z lines. Remember that also. Okay, that's all. And corpus callosum connects two cerebral hemispheres. Thank you, children. Thanks a lot. Bye-bye and all the best. You are going to give your best. I am going to pray for you. Stay calm. Okay, do not be worried about anything. And yes, that's all, children. Go. And do not ask around everyone whether they have studied everything or not. If you have time, just go through whatever you have studied now. Okay, that's all. And one more thing, ethylene acts as both growth inhibitor and growth promoter. Ethylene is a hormone that acts as both growth inhibitor and promoter. Okay, and that epigynes thing, I'll just tell you now. Yeah, epigynes is basically when it is towards the lower part. That is epigynes, and perigynes is basically when it is like in the equal level. Okay, and hypogynes at the top. That's all, children. Bye. Maths would be coming. Your answer will be there at 4. Okay. Bye. Take care. I'm ending the live. Okay.